Hello everyone, my name is Veronica Nash and I am the US Regional Head of Flow Cytometry at Cerba Research, which is a controlled research organization that offers general and specialty testing and support of clinical trials. Today my talk is going to be about our work on Cytek Aurora instrument standardization with the purpose of using them for global clinical trials. For the purpose of the talk, I will give a brief introduction about what clinical trials are and how flow cytometry supports them, and then we will dive into how Cerba achieved the monitor standardization of Cytek Auroras in multiple sites. And then after conclusions, I'm going to be very happy to answer any questions. Clinical trials are the processes where a new developed drug has to go through from its discovery to its approval on the market. Every single phase has a particular function that goes from the determination of safety, dosage, evaluation of toxicity, benefits, and the effectiveness of the drug for the indication or disease it was created for. Flow cytometry supports all of these phases of the trials with a variety of assays. So they can go from just enumeration and characterization of cell of the immune system or therapeutic cells like CAR-T. The flow cytometry can give information about kinetic of the drug with receptor occupancy assay, which kind of cell are activated with intracellular cytokine staining, and more in general, it gives information of characterization and monitoring of biomarkers that are defined as those markers that are associated with a particular indication and whose change in expression can give you an idea and a lot of information on about the effect of the drug for a particular indication. So clinical trials are global studies. Patients are recruited from all over the world, and it is very, very crucial that the samples arrive in locations that are located in crucial areas where the sample can arrive with stability. So that is why it's so important that these locations offer assays that are the same everywhere. And for these assay and testing results to be considered the same, the assay needs to be standardized, which means that it doesn't matter who is performing and where the test is performed, the expected results must be the same. So the Cerba Research last year purchased two spectral flow cytometry to add to our already standardized fleet of conventional flow cytometers. And this was to respond to the increasing needs in clinical settings of gathering more information and a deeper characterization of a patient sample coming from the same tube. This also fulfills the need of discovering possible new biomarkers and also unexpected dynamic in the immune system. And this, the most important is that all of this amount of information can be collected from samples that are very precious, as for example, bone marrow aspirate that most of the time arrive to the site in very, very small volumes. So the challenge and the work that we did was then to implement and standardize our instruments in US and in EU. So, of course, before you get to the standardization of your experiment, you have to make sure of your instruments. You have to make sure that your instruments are functioning properly. So you have to make sure, so your field service engineer makes sure that the instrument is working according to manufacturer specification. And then you have to perform your performance qualification, which is a series of tests where you make sure that your instrument is performing consistently over time for its intended use. And then after all of this is, sure, you go ahead and you perform your instrument standardization. So the first thing that we did was we defined what was our predicate and our comparative instrument. We identified the instrument in US as our predicate, and the first thing that we did was to collect baseline MFI target values. We did that by running SpectroFlow QC beats that are the QC beats provided by Cytek to run every day, but in an acquisition module. That way, we recorded all of the MFIs coming from for every single detectors, and you can see them plotted in this graph. So these were our MFI values for our predicate instrument at baseline. Next, we did the same exercise in our instrument in Europe. We ran the same lot of beads and the same lot of SpectroFlow beads in the acquisition model, and we checked the variation of our MFI target values between the two instruments. We requested Cytek to have an acceptance criteria a little bit stricter than usually it's considered, like standardization of instrument that was of 5%. And we were very, very happy with the results. The instruments were performing already like very beautifully. If we overlap the two values, the two MFI set of values, they are basically like overlapping. All the variations was below 2 and 3%. So we were very, very happy. So next, we decided we compare fingerprints of the instrument. So what do we define in the fingerprint of the instrument? We generated a spillover spreading matrix by staining freshly thawed PBMCs for CD4. We use a kit coming from Cytek, which provide a set of antibody that are excited with fluorochrome, that are excited with the violet, blue, yellow, green, and red laser, and we bought additional 5 antibody from BD for the UV laser. So we stained the PBMC, we recorded MFI, a mix, and calculate the matrix with the help of Cytek. And basically, this matrix will give you an idea of the spread that you collect of like this fluorochrome in all of the other detectors, and this is a fundamental information to just characterize your instrument for your panel design. So, but what we did was then we thought, like, if our instruments are comparable and standardized, they should also have this fingerprint comparable. So we repeat the same exercise also in EU, and we generated the map. And as you can see, they really correlate very nice. So visually, the spread is really like, it's in the same spot. We have some differences in certain areas, but when we calculated the delta matrix, we are within a delta of 10, which is in accordance to Cytek observation of the definition of instrument comparability. So after this, we wanted to really see if our instruments were like having really differences or not. So what we did was we tested a biologically relevant assay, but we tried to remove variables that were not specific just for instruments. So it's we use a lyophilized kit of PBMC pre-stained with 18 markers, and we run the same lot of the kit into different locations. The only thing you have to do with this kit is just adding some buffer, same amount in both locations. And so we expected that any difference that we would observe in the results was coming just from differences in the instrument. So, not to our surprise, the two instruments were beautifully performing. So when you compare just visually, like the plots, it's just like you can't tell which plots are coming from one instrument or which ones are from the other. And I just want to remind you that this is of absolute importance when you're running clinical trials, because you don't want to have any variation that is coming from where you're performing the assay. So this is exactly what we were expecting. If we dig more into the data and we compare the frequency of the populations, you see that they are very, very comparable. And then if we go more into the statistics and we compare percentage of difference, where the numbers between the results from our predicate and the comparative are compared with this calculation, we see that everything is below 20% of difference. It is our criteria, except for the populations that are defined as rare, below 5%, where you would just expect more variation because of the math. And next, we also compare MFIs coming from the same assay into two different instruments. And I think this is probably even more striking, as like they basically just beautifully overlap. The only difference that we see, it's a little bit in the negative of these two markers and these two colors, but I think that we didn't expect that this was going to be this good. So when we compare the also MFI's values and percentage of difference, we see that we are beautifully below 20%. So in this, we thought that our job was done and that our instruments could be considered standardized. So the final test was, okay, what if we then try this in an assay that we would normally run for clinical trials? So we validated an assay coming from a 14-color kit from Cytek, which gives you like a pretty good already characterization of your immune system. We use PBMCs and we had to do some development for use of reference control, titration of viability dye, and we validated this in US according to CLSI H62 guidelines. We transferred the assay to Europe, so all of the protocols, process, acquisition, template, settings were transferred to our lab in EU, and we performed a correlation experiment. 3 PBMCs donor were shared between the two sites, and they were both in both sites. They were tested in triplicates, and then the run from one side was compared to the run from the second side, and the percentage of difference of the results was calculated. And this is what it looks like. Again, the output is highly comparable, really nicely correlated. And when we look at the data, we are below our 20% difference, which is what we expect when we run clinical trials. So we cannot have anything different than that if we want to run the assay in globally. So our sites are ready to run, and with this I have my conclusions where I showed you that the reproducibility of the data across different points and between instruments requires proper standardization. We define a process for Cerba Research to achieve the standardization in the instruments located in US and in EU. And I think that with our work, we demonstrate that with Cytek Aurora, even though you increase complexity in your panel design, the advantages that you have by using these instruments are like so many, and you can still use them in global clinical trials reliably, and have the advantage that you can use, you can have so much information from limited sample that are available. With this, I want to thank my group. This is the Cerba Research scientist group. And then my counterpart here, Jan Spitals. I suggest if you want to learn more about what we do for performance qualification to go check his poster. And then, of course, I cannot forget to thank Cytek. They were absolutely fantastic in support of this, especially Ame was. We had many discussions. We learned from each other. It was an absolute great process. And if you want to learn more, please, a little bit of self-promotion. June 7th, we will talk more into details about clinical trials and how to, set up panels and stuff. So you can register here. And now I'm ready for questions.